So going to process development and HCP characterization. So one of the first steps in the holistic controller strategy was to obtain knowledge and understanding of the levels of HCP, but also what type of HCPs were in our map production process, and what were the HCPs that were remaining in the bulk drug substance in the process. So for that, we use characterization of the HCP profile by LC-MS, and the analysis was performed on in-process samples for clarified harvest, a chromatographic step 1, a chromatographic step 2, and the bulk map product. So here you can see that we performed the LC-MS analysis, and here are the identified HCPs plotted in a 2D plot where we have the PI on one scale and the molecular weight in the other scale. As you can see, there are many HCPs in what we call the 4 quadrants of a 2D plot. What we can also see is that we obtained approximately 800 HCPs were shown in the clarified harvest. About 72 of them, the ones marked in blue here, were identified in the first chromatograph after the first chromatographic step. And then about 4 were identified in the second chromatographic step, that the the green ones. And about 3 were identified at last in the bulk sample. You cannot see them all here, but they are around PI of around 8, and it's consistent with our products PI. It's around also the same area. So there were only 3 proteins left in the bulk. One of them was identified actually as a residual protein A from the chromatographic column 1, and a second of the third was a problematic HCP. So we tried to investigate a little bit further on that, but we also could see that we have a nice reduction through the process. One thing I would like to say is that knowing the PI or molecular weights of the proteins, HCP proteins, it aids in developing a purification process in order to remove them.